Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there.
Hello everybody, welcome back to another episode of the A-Level Cookbook. Today we're going to be doing the OCR A-Level Biology A, June 2019 paper. So let's get started, paper 2. So which of the following is more likely to increase biodiversity? So biodiversity is all about having large variety and variability of like different species and stuff. So you've got lots of different species and different numbers of each species. So whichever one of these options is going to be it. So captive breeding programs can do that because if you've got something that's near extinct or whatever, you can start like, captive breeding them and things like that and bring them back up in numbers. That's definitely one that can. Climate change is reducing it. Human population growth is also reducing it because, we're getting rid of like land and stuff for the things we need. And monoculture is where you're growing all of the one same thing, so that's immediately reducing it. So by proxy, that's got to be A. A teacher wrote that a garden is a dynamic pond that is home to a variety of organisms. The temperature of the pond varies depending on the weather and the time of year, and this affects the populations of the species that live there. Which of the following terms applies to the teacher's description of the garden pond? So if we just look at each word, a community is all of the populations of different species in a habitat. So like every single one of the rabbits and like squirrels and whatever living in like the woods. An ecosystem. So an ecosystem is all of the organisms in a particular area and it's non-living or abiotic conditions as well. So like you take a community and you slap the abiotic conditions in, that's what that is. Now a habitat is where an organism lives, and a niche is the role of a species within its habitat. So what it eats, when it feeds, what it interacts with other organisms, what it's eaten by, what it eats like other organisms, and also where it lives and when it's active. So whichever one of these terms accounts for the fact that these varieties of organisms are changing due to biotic factors or abiotic factors. In this case, the best thing that would account for it is B, because we're looking at a variety of organisms, and what the variety is really depends on the time and the temperature. Biodiversity is important for the new development of medicines. Which of the following statements about the development of new medicines is incorrect? So computer modeling can be used to identify useful medicinal compounds. That is true, so that one's already out. Genetically modified bacteria can synthesize medicines. I mean, we use bacteria to make insulin, so that one's out. Many pathogenic bacteria have become immune to antibiotics. When they don't become immune, they become resistant. So that's that one, because immune implies that they're making their own white cells and stuff and going and attacking it and this and that and antibodies, and they're not doing that. And microorganisms are an important source of new anti-medicines. That is true, because we got penicillin from fungus, so that one's got to be C. So just remember in these questions, the phrasing and the terminology also matters, not just like what fact it's conveying. Hox genes contribute to the overall body plan of an animal. Which of the following rows describes Hox genes? So body plan is just a general structure, which is how your body's laid out, where your arms and legs go, and the proteins that control that are encoded by Hox genes. Hox genes contribute to the overall body plan of an animal. Which one correctly describes Hox genes? So body plan is a general structure of an organism where its arms and legs go, right? And you have these genes called Hox genes. Within these Hox genes, you've got something called a homeobox sequence, which is like a region that codes for a specific protein called the homeodomain. This then acts as a transcription factor, so it can either enable protein transcription and translation or inhibit it, which leads to the changes that we see in these organisms' layouts. Now, the other thing, though, as well, is that these homeobox sequences are quite highly conserved, meaning that they've not changed very much during the evolution of different organisms. So they're quite similar. So, for example, like between like monkeys and humans, our body plans are quite similar. So in this, we're looking for that which fits that, basically. So here I'm looking for things that I can immediately knock out. So already I can say that, well, I mean, Hox genes make a protein which then forms the homeodomain, which can then bind to it. So it's not really the whole thing that's a transcription factor. So these two are out. And then between these two, similar animals, that's correct. They both make a polypeptide. But mutations are often lethal. Like things just don't develop in the right ways in the right places. So that's immediately the one that's correct. Little to no effect is definitely not the case, considering these are, where your body plan is. Hox genes contain a homeobox sequence of 180 base pairs. Two species have a homeobox sequence of 180 pairs, where 1.7 of them are different. Sorry. Which of the following shows the number of amino acids coding for that would be different in these two species? So we have 180 base pairs, right? So that's 180 that would be in the sequence, and 1.7% of them are different. So what we do is we work out 1.7 of that as a percent. So we're going to get, if we do that, 3.06. So the minimum has got to be, 0, and the max is 3. So it's got to be B. It doesn't matter that they're in pairs, because if it's this, this, and this, and then this, this, and this, they're just going to be complementary base pairs. So this triplet's what's read to actually then go and be transcribed and translated. So meiosis is an important factor of sexual reproduction. Which of the following processes occurs during meiosis and contributes to genetic variation of offspring? So in meiosis, we get crossing over. Gene mutation is random. It does not happen during meiosis, and random fertilization is after meiosis. So it's got to be just one, which is D. Random fertilization is only when the sperm and egg have actually been made and gone and done their job, like fusing. That's not during meiosis. And mutations are entirely random, and it's not like meiosis is actively making them happen. The adult wandering albatross, this has a wingspan that ranges from 2.5 to 3.5. Which of the following describes the variation in wingspan by wandering albatross? So if it's got a varying length of characteristics, like, you know, like for example, the height or width or something, then it's going to have an environmental impact as well by default. Otherwise, if it was in purely a genetic thing, if they had the gene, they had the thing. So if there was a gene that made you 1.7 meters tall and you had that, then you'd be 1.7 meters tall, right? But those that are 1.7 don't have a gene that says, hey, you are 1.7 meters tall. In the same way that some people are taller and shorter and so forth. I'm like 1.78 meters. So like, you know, others might be 1.6. So there's variation in that, and that's because of environmental factors. So immediately we can cross you out. And the polygenic bit as well, because there's still going to be environmental factors playing a role here, because there's quite a lot of variation there. So that leaves us with B and C. So it says interspecific and controlled only by environmental factors. Well, it can't be. It's got to be both genetic and environmental, so it's C. There you go. Which of the following is not associated with the use of artificial selection in farm animals? So artificial selection is the same as selective breeding, which is where you get a few individuals in a population and you find that they have advantageous characteristics and you breed them. For example, you know, like, I don't know, like cows that make a lot of milk. So now it's saying which is not associated with the use of these, right? So when you're doing selective breeding, what you're doing is you're breeding closely related individuals, and that's called inbreeding. But the problem with that is that you can have a limit in the gene pool, which can mean things like recessive disorders and stuff are passed on and things are then expressed. But as well as that, you can have things like, you know, like health problems and stuff in that sense. So then if we look at what's not associated, that is associated with it, so that's out. Inbreeding is associated, so that's out. Increased frequency mutations isn't really associated with it. Again, at random and just, it's not like we're putting radiation on them, so that's one of them. And a reduced gene pool is associated with it, let's see. Which of the following is not a valid concern about the use of genetic modification? So this could be a problem because it's genetic, you know, antibiotic resistant genes. Same for herbicide resistance genes as well. Certain seeds might not be able to be used by poor farmers. Again, yeah, because like if poor farmers can't afford to get this nice little fancy genetically modified, like, you know, wheat plant or something, that's not going to be a good idea. And the use of human embryos in stem cell production is unethical. Well, that's relating to stem cells and not artificial genes and gene modification, so it's D. And that question seems a bit goofy. Barnacles are small animals that live on rocky shores. Adult barnacles are fixed to rocks and do not move about. They catch passing food in the water with modified limbs called cirri when the tide comes in. This species is a barnacle found in UK shores. Two students wish to estimate the population size on a rocky shore near their school. Which of the following could they use to estimate the population? So in this circumstance, we need to figure out what kind of thing we're dealing with. If we're dealing with a, you know, like a moving thing, then you're going to have to use mark, release, recapture. Whereas if it's something that's non-mobile and moves very slowly and sits there, then you're going to use the other one, which is like quadrat sampling and stuff like that. So immediately, mark, release, recapture's out, and quadrat sampling's there as well. We want an abundant scale, because if we use an abundant scale, it means we can work out how many of them there are, how many population there is. Abundance is working out how many there is. Distribution is where they are found. So in this case, it's 1 and 2, so it has to be B. So corals are a group of animals that usually live on the seabed close to the surface of water. Many corals can reproduce sexually and asexually. Which of the following statements about asexually produced coral offspring is not true? So all offspring produced from an individual will be genetically identical. Well, that is true because they do mitosis to do that. If a change in the environment harms one of the offspring produced from an individual, then it'll probably harm them all. Yes, because they're all clones of the original thing, so that's out. Meiosis occurred in order to produce the offspring. That's the one that's incorrect because it's not meiosis to do asexual reproduction, because the whole point of meiosis is to make gametes for sexual reproduction. So that one's out. And then the offspring will tend to thrive if the conditions are similar to those present with the parent organisms reproduced. Well, again, they're clones of the original thing, so no. So it's C. So it says, which of the following nucleotides contains uracil? So uracil is an RNA nucleotide, right? So it's going to have ribose as its sugar, not deoxyribose. So the way to tell is that you look at the sugar, the pentose sugar, and you look for whether it's got 2 OHs. If it's 2 OHs, then that's ribose. Whereas if it's only got 1, because there's an H here instead, this is deoxyribose. It's missing an oxygen. So because it's RNA, not DNA, all the deoxyriboses are out. So you two are gone. Now we need to decide between these two which one it is. Because uracil is replacing thymine, we're looking for pyrimidine. So pyrimidine only has 1 carbon-nitrogen ring, right? Whereas purines have 2 that are joined together. This one has 2 joined together, so that's a purine, so that's out. So the only one it can be is A. The ability to roll one's tongue is under the control of a single gene. The gene has 2 alleles, big R and little r. People who can roll their tongues have the genotype big R, little r. People who cannot roll their tongues have little r. A survey showed that 12% of a population in a school cannot roll their tongues. So that means little r is going to be 0.12 as a decimal, which is 1, remember, as a total, which is 100%. The students then use the Hardy-Weinberg principle to calculate the number that heterozygous in the school. Which one represents the heterozygous? So when using the Hardy-Weinberg equation, you need to define what you're going to call P and R. So I'm going to say P is my big dominant one here, and Q is going to be the little one, so little r. Did I say p and r? I meant to say p and q, sorry. So in this case, if you're unable to do it, your little r, which is 0.12, right? So that means that big R, or big R, little r equals 1 minus the 0.12, as in the rest, which is 0.88. Now, the problem with that is we don't know how to divvy up the big R, little r, right? But that's where these formulae come in, and that's why calling these things help. Because if we look at RR, that's the same as saying QQ in this circumstance, which is just Q squared, which is 0.12. So that means Q is going to be the square root of 0.12, which is 0.346. So if that's the case, then we can go one step further. Or if we know what Q is, we know that Q equals 1. So 1 minus Q equals P. So P is going to equal 1 minus that answer we just got, which is going to be 0.65356. Let's go with that, just to round it. Okay? So then we know what P and Q are. This P squared is going to be, what, big R, plus 2PQ, which is big R, little r, or little r, big R. That's why it's 2PQ, because they're the same thing. And then Q squared is going to be little r. So we're looking for the 2PQ. So 2PQ is just going to be 2 times the P, which is what, 0.6358 times 6, times 0.346 times 2. So if we do that, we end up getting 0.452, which is roughly 45.2%. So that means the answer to this one is whatever fits that the closest, which is indeed D. There you go. So now it says which one's not involved in the primary response against infection. So primary means it's in your body for the first time, never been seen before. So immediately memory B cells is out because you don't like where they're coming from. Now this image shows a diagram of an enzyme. Here's your active site, here's an allosteric site, and it says which of these could be a competitive inhibitor of this enzyme. So it's whatever's complementary to the active site. Can't be the allosteric because that's a non-competitive inhibitor. So immediately, the one that we're looking for is whatever fits here, which is D. So it's D. There you go. So now it says define osmosis. So it should be 3 marks for you guys at this point. So it is the net movement of water across a partially permeable membrane down a water potential gradient. Plants rely on osmosis for support. Explain the importance of osmosis in plant support. So we're talking about support as in physically holding them up. So what happens is you've got a vacuole in the plant, and the whole reason for that is because if I draw this plant cell here with a vacuole in it, like it's all, saggy and stuff, when it fills up, it's like this and squishes and pushes outwards. And that causes something called turgor pressure, which keeps cells turgid and helps them stand upright. So that's what we're going to say. So we need to say every single step from here to here. So you can't just say osmosis gives them turgor pressure. You need to say by how, step by step. So water enters the vacuole of plant cells via osmosis. Then what this does is it increases the vacuole size and pressure against the cell wall, because it's now squeezing against that cell wall, pushing it, trying to get out against cell wall. This leads to an increase in turgor pressure, and these turgid cells support the plant. So step by step here from start to finish, and you'll never miss anything. So the apparatus shown in this figure can be used to demonstrate osmosis. So what we have is we've got a level of solution X, and now we've got a capillary tube, solution Y, a selectively permeable visqueen tubing, then solution X. Fine. When the capillary tube with visqueen tubing bag was placed in the solution Y, the level of solution inside this rose from here to here. The ruler used to measure the distance was accurate to the nearest 0.5 millimeters. Calculate the percentage uncertainty. So percentage uncertainty is the uncertainty over what value you got times 100. Now the thing is though, we need to work out what our raw uncertainty is. So they've measured it from one place to another. So we have 2 lots of 0.5 as our uncertainty. All right, because they've read it twice. So they've got to double the amount of uncertainty in that. So that's going to be 1.0. Now the actual value that they measured is the difference between these two, which is going to be what, 16 millimeters, right? So then to work out the percentage uncertainty, all you do is 1 over 16 times 100. And if we do that, we get 6.25. There you go, 6.25%. Then it says, what conclusions can be drawn about the composition of solutions X and Y? So if we have a look in this question, it's told you that inside the tube, the capillary, the volume had gone from 10 millimeters to 26.5. So this level of solution had gone up. So if it's gone up, then it means water must have entered this to push it all up. So we're looking at osmosis, remember? So the reason that it's gone up must be that the water potential in Y is higher than in X. So then you would say that. So Y has a higher water potential than X. There you go. So therefore water moves in by osmosis. So you have to make a comparative statement. You can't just say solution Y has high water potential. It has to be higher. And also as well as that, you can't really say hypertonic, hypotonic. That doesn't show them a comparative statement. What they want is that it has a higher than X, so water enters X via osmosis. So a group of students use the following method to investigate osmosis in plant cells. So always remember to do your independent and your dependent. So try and look at all the question just to quickly collect what it could be. So what are they doing? Well, they have got the independent is also, well, first of all, it's the plant. They're changing it, the actual species, and they're also changing the sucrose concentration for each. And their dependent is percentage change in mass. This might be useful for later, we'll see. So what they did is they cut plant material of equal surface area, ensuring no skin is present. They rinsed to remove debris. They planted them gently with a dry paper towel. They weighed them and recorded the mass, put them in a 200 centimeter cube beaker, and then they covered it with 50 centimeters cube sucrose solution and left them in these different solutions, left them for 24 hours, removed the piece of paper, the piece of plant material, dried it again and weighed it, and then calculated the percentage change in mass, and then repeated twice for each, and then here's the results for 3 different plants as shown here. Fine. It's just really helpful to have a quick look at what's going on, so we'll just have a look at the means to get a vague idea. So we can see that the mean percentage change goes, they actually get bigger, more waters get entering versus them losing it. So between these two concentrations is probably where the concentration of the carrot would be, where it's 0, right, where there's no movement. So here as well, it's going all the way out, it's increasing, and then down here at these concentrations. So again, these mystery concentration of these two must be between these there. And then for the same one here with potatoes, here, and then there's the change here. So then these two must be where, or between these two must be where the water potential of the potato is, because that's where it wouldn't be any movement. So now it says, why was it necessary to calculate the percentage change? Right, you can't just say to allow comparison. That doesn't really help. The reason we're doing percentage change is because they're going to have different starting masses. Because think about it, like potatoes are a bit more dense than, like, I don't know, courgettes, I think. So they're going to have different starting masses. But that means, using a percentage allows comparison. That's, you need to say why a comparison is allowable or needed. You can't just say to allow comparison. Why? Like, why can't I just use the same normal results? You have to explain that second bit. The students identified replicate 3 of the potato in 0.7 moles per deciliter cube sucrose as anomalous, suggest a practical error that might have caused this result to be anomalous, and then explain the likely effect. So if we look at the replicate 3 of 0.7, so 0.7 replicate 3 is here, what do we see? So we can see that these two have lost more mass than this one, so this one's not lost as much. So we need to just note down what could be a possible reason is. So it has a higher mass than expected, right? So you've got to link back to the steps on what they took to see what could screw up, making them have a higher mass, which would make them have a less percentage change in mass, right? So in that case, if I have a look at all of these things, the eventual weighing here, maybe they just didn't dry it properly, so there was more water on it at the end, so it looked like it didn't lose as much. Or you could have said it maybe had, like, maybe at the start they cut it from different you know, like places of the potato that had higher water potentials or different water potentials. So I think I'm going to go with my inadequate drying one, and that makes sense. So inadequate drying, therefore higher water makes it have more mass than the others. So it's falsely inflating. Use table 16 to identify which one had the highest concentration of sucrose and then justify your conclusion. So the idea here is that if wherever we have the changeover point, like I was saying before, where there is no percentage change in mass, that's where they're going to be equal, the water potentials of the two. If the water potentials are equal for 2 of them, then it's pretty alright to assume that the sucrose concentration would also be similar. So in that sense, we're looking for whichever ones have the highest change, basically, like bit where they change over. So here and here, there's the changeover between 0.3 and 0.5. Here and here, here and here, it's between 0.3 and 0.5 as well. Whereas with the potato, it's between, 0.1 and 0.3. So these two could withstand a higher sucrose concentration before they started losing water and dropping in mass. So either one is fine. I mean, I'm going to just pick the carrot because I can't be bothered writing courgette. So the carrot, why? It's because the 0% mass change lies in between 0.3 and 0.5 mole decimeters minus cube, right? So this is higher than the potato where there's no change. And then as well as that, we need to still explain why it had the highest sucrose. So if we look at the other concentrations, if I look at carrot here, it's got minus 6.23 here versus courgette, which is what, minus 4.27, and the potatoes is minus 5.7. So it's actually lost the most at 0.7, which means very little water has actually moved out as well. So for courgette at 0.7, it lost the least mass compared to the others as well. If we go a bit further to narrow them down between the carrot and the courgette, if we can, if you look between these two, so that, and then here and here, you're going from plus 1.43 to minus 2.27. So the jump is pretty big, like in this, the difference between these two is a lot higher than this. So here you can see that we're losing minus 2.27. So if I was almost to draw it like a graph where it's like, you know, the percentage changes on the y-axis, here we are, right? So here and then here, it goes down more steeply from there to there. Whereas with the courgette, Like, it might start like up here and then get like this. It's a little bit less like drastic, the drop. It's a really hard way to draw it, but almost like that, or like this, I guess. So if the drop is less drastic, that means that this has a higher concentration at which there is no mass loss. So because you can see again, it like becomes negative at 1.23 at 0.5 versus this one, which is already overshot. So the courgette has a higher concentration at which 0% mass loss is. There you go. So water has many properties that are essential for living organisms. Explain how these properties relating to the density of water contribute to the survival of these organisms. So you can't just go and yap about everything else. You need to talk about the density. So the key fact about density to do with water is that it's less dense when it's a solid and it floats. Ice floats, right? So if ice is floating, you need to think of a use for that in biology. And the first thing that I'm thinking of is it's floating on a river, which is useful because, well, let's start off by saying ice floats on water because it's less dense. Why is this useful? Because it can insulate the water below. So that means that, you know, if you can do that, then it can also mean that this can provide species habitats below and also within the ice as well, and on the ice as well. Like, think about polar bears, what they're living on, right? And then, as well as that, what else can we say? Well, if you're thinking about like fish and stuff like that, for example, if they're able to float, it means that the density of the two things must be similar. So water density is similar to living organisms, so they can float. Otherwise, they just sink to the bottom and that'd be very useless. So the fact that they're able to float and be buoyant is because they have similar densities. DNA could be obtained from a variety of plant animal cells. A group of students tried to purify some DNA from leek cells using this following method. They decided that the exact volumes were not necessary. So they grind a leaf leek to fine pulp, and then what they do is add salt and cold water, mix again for 10 seconds, add protease for 10 seconds, filter the liquid into a test tube, and then stand for 10 minutes. Then tilt the test tube and add some ice-cold ethanol, and then a white layer of DNA forms between them, and then extracts the white layer using a glass rod. So they say, what's the point of step one? Well, they're just grinding it to a fine pulp. So the whole point is just to break down, break cell walls. Suggest why protease enzyme in step 3 is needed to purify DNA. So you need to think, proteins and DNA, what could it be? With the protease, it's an enzyme that breaks down proteins. We're looking at trying to get DNA out. So it must mean it's breaking proteins with DNA. So it must be that it breaks or hydrolyzes histones associated with the DNA or around the DNA. There's no proteins in the DNA, it's just that there's histones around it. The student considered using pineapple juice as a source of protease enzyme. Suggest why this would not be an appropriate source of protease when attempting to produce a pure sample of leek DNA. So, I mean, well, first of all, pineapples are also, organisms that are living things. So there's going to have pineapple juice could contain DNA. Another thing you could say is that it's quite acidic as well. So it might be that the pH is too low and that can mess up with a lot of things in the DNA too. So just one important step that the students had left out of their method. So, well, one big thing is that they forgot to add a detergent, because if we have a look, the reason we added detergent is because it breaks down cell membranes as well. So yes, the grinding and this and that will obviously break down the cell walls, but cell membranes may still be intact. So by doing that, it breaks that and actually releases all the goods. So we can't really do very much other than that. Then it says, describe what step 6 says. So step 6 says a white layer of DNA is formed, that is precipitation. So genes isolated from DNA can be used in gene therapy. Cystic fibrosis is a disease that can be treated with gene therapy. Healthy individuals have a gene that codes for a channel protein called CFTR found in the plasma membrane of a variety of cells, including those of the airways of the lungs. People suffering from CF have 2 copies of recessive allele, so their cells do not make the right channel protein. The allele that codes for the functioning CFTR protein can be inserted into the DNA of CF sufferers. The cells can then synthesize the correct CFTR protein as function as normal. The treatment of cystic fibrosis is known as somatic gene therapy. Another type of gene therapy is germline. Complete the table to show 3 differences between somatic and germline therapy. So somatic means, soma means body, so body cell therapy, and germs are gametes. So you need to compare the differences. Now you can't say one is legal, one is not. That's not the differences between them. And again, nothing to do with ethics. They want to know, biologically speaking, what the differences are. So immediately, somatic is in your body cells, because it's going to be in the lung ones only, so it can't actually be passed off. So not inherited. So it can't be passed on to offspring, right? Now, as well as that, where's this going? This is into body cells, this is into gametes. And then what else happens as well? Well, all cells in this germline will have the functional gene. Whereas in this, it's only body cells have the functional gene, like the ones that you've inject, put them in. As well as that, this is short-term and temporary, and you might have to repeat it non-stop because cells obviously die and reproduce and this and that, blah, blah, blah. Whereas this is long-term, you don't have to repeat it because it's there forever. So some attempts at gene therapy have resulted in change to the functioning of the other genes. Explain how inserting a new gene into a chromosome could affect the functioning of other genes. So a gene is something that codes for a protein. It's essentially saying what's screwing up the expression of another protein. So what might be happening is, imagine I've got this nice sequence, 1, 2, 3, 4, and then I just slap this in, and then there's that, right? Imagine we have a sentence that says, I don't know, sad cat rat, right? And then I just add 2 extra letters in, And you can only read in threes, by the way. So now I end up saying, I don't know, let's say we slap in a G and a C. S, A, SAG, C, D, C, A, T, R, A, T. Can you see how the whole reading frame has been shifted? It now says SAG, C, D, C, A, T, R, A, T. So that's what's happening here as well. You can't just slap a thing in and be like, oh, this is lovely. It's going to screw up everything else. So now we've got something called a frame shift, which means you're going to alter downstream, which just means further down, triplets. Therefore, this could affect transcription and translation. Or it could maybe affect, like, switching off and on genes, or it could be that it actually could disable a functioning gene if you slap it right in the middle of one. CF occurs when individuals have two copies of a recessive allele. Huntington's disease is a lethal disease caused by a dominant allele that codes for the Huntington protein. So just why gene therapy is unlikely to work as a treatment for Huntington's disease. Well, it's because it's dominant. So, if you put gene therapy into this and try and get like one copy, if you've still you can only replace one really. The whole point of gene therapy is you kind of replace the recessive one with a dominant and you're chilling. In this case, you'd have to try and do both, and it's kind of difficult to do that. So in this case, suggest why it wouldn't work. It's because Huntington would still be produced. The Lake District is the largest national park in England, and it covers this area. It has a wide variety of species, some of which are under threat or endangered, and the resident population is about 41,000 people and humans. In 2016, the Lake District had this many tourists. The proportion of Lake District land for use for different purposes is shown here. So you see, there's a lot of different things going on. It says, explain one way in which tourists can increase the biodiversity in an area. Well, if you think about it, if there's a lot of people who are like, oh, I really like the fact that there's lots of different varieties of organisms and habitats here, lovely, then obviously, because they, people these days want to maximize that profit incentive. They want to get those big cash payouts. They're going to be like, all right, you know what? We should probably not endanger all these animals and actually kill them all off. Maybe we can get some money from this. So in that sense, the money from tourism could support conservation. As well as that, you've got ecotourism. So by keeping lots and lots of varieties in this sense, they're keeping all of these animals and their habitats nicely, chilling and stuff, and keeping the biodiversity good. Just like every other thing in the universe, you gotta have a profit incentive to it. The Lake District contains large areas in which timber is produced. One of the aims of the management is to produce timber sustainably. Estimate the percentage of land that is covered by woodland and forest. So if we have a look at this diagram, then we have this going on. What we've got to do is we've got to use this figure to have a guess what's going on. So if you look at woodland and forest, it's like this here, right? Which is basically how many of those can you fit in this? Ideally, you should measure it with you should measure the angles, but like 1, 2, 3, 4, 5, 6, 7, 8 almost. So it's an eighth of the whole thing. We could try going with that. So then 1/8 as a percentage is what? 12.5, 13%? Timber could be produced economically by a technique called clear felling. Clear felling can damage biodiversity. Explain how it's possible to produce timber sustainably using clear felling. So clear felling is where you're sort of cutting down trees in an area, like all uniformly in an area of forest. Now here it's saying, how can you do this and still have sustainable timber production? So clearly what they must be doing is they must be limiting, first of all, how much they're doing. So they're limiting the area felled, because you want to still give them a chance to just chill, And then as well as that, the whole point of sustainable is that future generations can still have access to this material or resource, right? So if they're chopping down all these trees, they must be replacing them as well, right? Replanting or replacing somehow. And then as well as that, other things they could try to do is they could try and have fixed distances between the replanted trees as well to make sure they're actually growing properly. That also means that there's less competition between them. Other things you can do as well is to try and minimize soil erosion too and things like that. But here I'm going to just stick with those two. A traditional timber production process that's still used in the parts of Lake District is coppicing. Describe the process of coppicing and explain the potential benefits of coppicing to the biodiversity of a woodland. So coppicing is where you're cutting a tree trunk close to the ground, and what happens is from that cut surface you can have shoots that start to grow and they mature, and eventually they're cut in place, and then you get more and more and more, and those shoots are quite useful because you can use them in other bits and bobs, right? So usually what happens is those shoots grow, and then about 7 to 20-ish years you start to get a coppice that's ready to harvest again. So when you're doing this, you keep repeating this process over and over and over again until you reach the trees that were first coppiced to begin with, and they will have grown to the right mature size, and you repeat the cycle over and over again. But the good thing about coppicing is that if you do it properly and you rotate and do it rotationally, then you maintain the biodiversity because the trees never grow enough to block out light. So you can't get successions. It's quite good because it actually limits succession as well in that sense, which means you're keeping a nice amount of biodiversity going on. So talk about that. So that we need to talk about how the process goes first, because that's what they want us to talk about, how it's done, and then we can talk about the benefits. So the actual process is what you do is you cut a tree trunk close to the base or ground. Then what happens is several new shoots grow from the cut surface. Then what happens is you protect them from grazers and things like that, so that it stops getting them eaten from grazers. and then you allow them to grow for a certain time, and then you repeat the process. And then what happens is you get, you're able to rotational coppice, which is where you just cut them, on a rotation, like literally just like bit by bit. And this means you can like repeat this basically forever. So repeat indefinitely. This is not talking about benefits. I'm just talking about the actual process first. Then in terms of the benefits, one, well, the other option is to plant whole ass saplings. Now, if you've already got these shoots and these stems growing, they're going to grow much faster. So these shoots or stems grow faster than saplings. So it means you can actually have more time for tree living versus dead. So that means lifespan of trees is longer. And then I was saying before about that biodiversity maintained because succession is limited because there's less shadowing of light. As well as that, comparing it to other options, if you do things like, for example, like felling and stuff like that, you can start to screw up the soil because it starts to get susceptible to erosion, because the trees are nice because they really bind the soil together and they anchor it with their roots and stuff, right? And as well as that, they remove water from the soil, so it's not too what's the word? It's like sandy almost, and particulate. And also it keeps me, because they do the nitrogen and carbon cycle, it keeps nutrient levels. So by doing this, because there's still things, tree bits there, it's holding the soil nicely there. So it prevents soil erosion and maintains its quality with nutrients. And as well as that, a nice other thing is you don't need large machinery either, so it's not going to obliterate the environment. So many schools visit the Lake District to undertake biology fieldwork. A group of students investigate the biodiversity of 5 herb plants they found in adjacent coppice and mature areas of woodland, and the results are shown in Table 18. The students calculated the Simpson's index of diversity of the mature area to be this. Use the information in the table to work out the Simpson's index of diversity for the area of the coppice woodland. So what we need to do is we need to use this formula, which is the sum of, sigma means sum of, n over n all squared. So we need to get every single n divided by big N, square it, and then add them all up. So we need to figure out what big N is. Big N is the number of all individuals of all species, right? So in this sense, our big N in the coppiced area is this. So N is 70. So what we're going to do is we're going to put a separate column for N over N squared, like this, and then work them all out. So for example, here it would be 35 over 70 all squared. We need to do that for basically every single one of them. So I'm going to work that out now, and then, yeah. I'm going to do it all in one go in my calculator, but you literally just do that for every single one. So for example, this first one would be 0.25 and basically the rest. So I'm just going to do that now and get what the sigma over n squared should be, just for the sake of time. So if you do all of that, lengthy calculation, you get 0.3506. So then D is going to be 1 minus your answer that you just worked out, which we got was 0.3516 or whatever, sorry, 3506. If I do that, we end up getting 0.649. We need to do it to the same number of significant figures in the question, which is 2, so that's going to be 0.65. There you go, woohoo! So now it says, use the example of the student's fieldwork to explain how biodiversity can be considered at different levels. So what that means is that if we have a look here, the index of diversity they got for this was 0.489, but we ended up getting it for 0.65, which are intrinsically different. So use the example of the student's fieldwork to explain how biodiversity can be considered at different levels. So the idea here is that when you're doing biodiversity, there is multiple ways of doing it. You can look at the species richness, you can look at the species evenness, you can look at the genetic diversity, you can look at the biodiversity index that we got, also like the habitats. There's different ways of comparing it. We need to use examples of their fieldwork to see how we can consider it at different levels, meaning what can we do to look at those different things, right? So we need to look at what the hell they actually did. So their fieldwork was these two, right? These two values, which is going to tell us our variety of species, right? Or species biodiversity. Because this is only biodiversity of species, right? So species biodiversity is from the Simpson's index, right? But there's lots of other ways you can assess biodiversity. For example, the habitats or species richness. So here, we can see there's 1, 2, 3, 4, 5 different species. So we can, here in their data, they've also found the species richness, which is the number of different species from their 5 plant species. As well as that, I mean, they've compared these two, coppices versus mature, which is another way of looking at it. So you can look at their variety of habitats as well. Obviously, we'd do more than just those and look everywhere if we really can. And we need to look again at using examples from their students. So I've said species diversity from their Simpson's index, species richness from the 5 species they did, a variety of habitats from the coppiced and mature woodland that they tested. But then the other problem, though, here is a critique almost, is that there's no way of really looking at genetic diversity from their work. No evidence of genetic diversity from their work. The question's basically asking, like, what are the other ways you can look at biodiversity, and what have these students done and not done, basically? Hey, bro, I got a text from this guy on my phone. I completely forgot I had him, bro. He's got saved as a tadpole. He was like, hey, bro, can you update my contact? I'm a frog now. So it's possible to clone animals using a technique called somatic cell nuclear transfer. So somatic means body cell, cell, nuclear transfer. Okay, so the most well-known example was the cloning of Dolly the sheep. 30 years before Dolly the sheep, successful cloning of an animal was carried out using a frog, Xenopus laevis. Frogs lay eggs in water. These eggs then develop and hatch into swimming tadpoles, and tadpoles grow a certain size to develop into frogs. So you got a donor tadpole, we just take their nucleus out, we take an enucleated, so that means taking the nucleus out an egg, put it in this, let it do its thing, and then here we go. Woohoo, we got a clone frog. Describe what's happening at stage A and suggest a practical procedure that can allow this to occur. So what's happening at stage A is that we're fusing just this nucleus and the enucleated cell. So here then, in this case, we're not really fusing like gametes and stuff, so be careful. And we're not really doing things like, just injection. That's useless. What we're doing is we're putting the nucleus in the egg. First of all, you need to describe what's happening. So nucleus is inserted or fuses into the enucleated egg. And then we need to say how. This could be via a needle, or it could be like an electric pulse, electrofusion, all of those kinds of things. Identify a key difference between the processes of stage A to C and the cloning of Dolly the sheep. So if we look here, we're putting them in an egg and it just does its job and it swims and does the other stuff. Whereas in Dolly the sheep, the embryo needed to be put in a surrogate mother. So embryo needed planting in the surrogate mother sheep. Whereas here, this just was free to start doing its job in the water. The frog produced in the process is not a complete clone of the donor tadpole, suggests why the clone frog might not be considered a complete clone. So one thing that I can say immediately is that we don't get our mitochondria from a sperm. The mitochondria that you'll always inherit comes from an egg, because the sperm can't get the mitochondria into the egg. So they'll have mitochondrial DNA from the egg. Another thing you could say is you could have random or spontaneous mutations that just happen. So now it says the success of SCNT has been investigated in many species. Sheep are more closely related to mice than they are xenopus frogs. Figure 19.2 shows the percentage of SCNT procedures that were successful in mice and xenopus when the donor nucleus was taken from cells at different stages of development. The xenopus data was in 1962, and then the mouse data was 1998. So we've got an early embryo, and we're looking at how it's changing over time for it's becoming an adult, and the stage of development of cells from which the nuclei were taken. So they took them from adults here, and then they took them from early embryos and in-betweens, and they said percentage of total nuclear transfers that resulted in a living organism, and we've got mouse here, and then we've got xenopus. So if we compare one by one, as you get older, the percentage that actually result in an actual living thing seem to drop across the board quite straightforwardly, whereas in xenopus, it's lower than mice, then it supersedes mice, and then it starts to go back down again. Fine. Suggest why the x-axis doesn't show the age of the donor nuclei. Well, I mean, it's because if you think about it, they're two different organisms. They're going to be living in different stages, different lifespans. That's one of them. It's not really easy to compare. So different lifespans, so it's not comparable. Therefore, they also mature, at different rates. You know, if they're like this age, for example, then they start making like sperm and eggs and stuff. It's not useful to compare the two of them because, the ages will look falsely different when in fact the, biological clock almost is like, just slower/faster, right? So that's that. Dolly the sheep suffered health problems throughout her life and died at an early stage. The donor nucleus that was used to create Dolly came from a sheep that was already 5 years old. The normal lifespan of a domestic sheep is 10 years. A student concluded that Dolly's health problems are caused by the stage of development of the sheep that provided the donor nucleus. There's 3 reasons why this does not support their conclusion from the figure. So they're saying that if you're 5 years old, the nucleus that you've taken means you aren't going to survive. So we need to use these data to suggest what doesn't support this conclusion. So already I can say that we have no idea whether they're actually, what their health is like. Literally all it just says is alive. So y-axis does not include the health of the organism. As well as that, we have one singular story that they're basing it off of, right? So it's a single clone. We need to say what the purpose of that is, the why. Therefore, the health problems may not be due to cloning, right? Like maybe, I don't know, he ate some dodgy grass or something and it was, things like that. As well as that, if we look at this figure, it's between mouse and xenopus. This is a sheep. So there's only mouse and a frog, xenopus. So it might not be applicable to sheep. As well as that, what year? I think in the question somewhere it said about the year that Dolly the sheep was like doing a job. 1996. These techniques were done in 1962 and 1998. So maybe the ones between 1962, for example, may, be very primitive and just a bit rubbish compared to what was done in 1996. And as well as that, correlation between them, surviving and being old or whatever, and actually not surviving, but actually living, and being older, doesn't mean there's a direct causation between them. There's no causative effect. So one measure of success in cloning procedures is the number of pregnancies that result in live births. Here's some information of the work of many scientists about the success of this in 4 different species. So goat, monkey, mouse, and sheep. Number of pregnancies, number of live births. But calculate the percentage of pregnancies that result in live birth in goats and mice. So in goats, it's going to be 8 over 26 times 100, which is 31%. And then in, mice? It is 56 over 438 times 100, which is 13%. There you go. So goats 31%, and then mice is 13. There you go. Enjoy. Compiling results from different scientists can have different problems, as their investigations may not have been controlled in the same way. Which factors could have been controlled when compiling the data to include in Table 19? So look at what they're measuring and what they're changing. They're changing the species, and they're measuring the number of pregnancies and live births. So we need to think of anything that could screw up either of those two measurements. So immediately what I'm thinking is the health of the mothers that the eggs came from, the stages of development or like of the surrogates, as well as that the conditions they were all kept in, where they actually fed well and nice and, you know, had space and this and that, as well as that the age, the question's also giving you this as well, like the age of the implanted embryo, nucleus donor, the age of the egg donor, and how many eggs they actually planted in a surrogate, because this doesn't tell us how many eggs they could. Maybe in this pregnancy, for one pregnancy, it took like 12, billion eggs or something. Okay, maybe hyperbolic, but you know what I mean? Right. And as well as that, what if they were also done at different times? So like the technologies got better and better. Those are some of the things you can mention here. So it's all about what's screwing up these two, right? So the control factors that I would say is health of the surrogates, the age of the nucleus donor, what else? And then number of eggs implanted. You can choose any of those things that I said, really, but those are the ones that I'm just going to go with because they're shortest to write. So plant cloning is often used by farmers to produce new plants. A plant that's often cloned by taking cartridges is lavender. A farmer had 2 fertilizer solutions, A and B, and they wanted to investigate which one to use on lavender plants in order to ensure the investigation would be valid. 2 cuttings were needed from the same plant. So it says describe how to clone a plant by taking a cutting. So what you do is you cut a shoot from a healthy plant, cut shoot from healthy plant, and you want to do this at a slant. Then what you want to do is you're going to put in rooting powder, so it actually, starts doing the job. So you dip it in rooting powder, put in soil, and like add water and all of that jazz. And then as well as that, you reduce transpiration by covering with a plastic bag. And the reason is because you want to keep it nice and moist so they're able to do its job of growing and photosynthesis and this and that, blah, blah, blah. And once it's strong enough to, be like, I'm ready to do my thing, then you can then go and take it to where you need to plant it for the rest of the job. So the whole point is almost like supporting it for the start in like artificial conditions and then letting it do its job. The reason we add it to rooting powder and things like that is to induce root formation. It's in the name, rooting powder. So it's all about just, that, really. The farmer grew one of the cuttings in soil fertilizer solution A and then one with B. And then they took several other precautions to increase the validity of this, including growing the plants in the same type of soil, exposing them to the same light intensity. After a certain time period, they measured the increase in the growth of the height of the lavender plants, and the farmer's results are shown here. So every single time, independent dependence. So the independent is whether it's A versus B for fertilizer, and the dependent is the increase in height. Cool. So we don't actually know what they start with or finish with. They just know that there's an increase in height. So now it says the farmer concluded that solution A increased the height of lavender more. A student said that even though the investigation was valid, the results did not give strong enough evidence to support this conclusion. Describe and explain how the investigation could be improved to have more confidence in any conclusions drawn by this, right? So all they did is they just did one thing and they say, oh, well, I know it's gone up 20 centimeters, that must be fine. But like, okay, but we've only done it once, dude. What if that was like pure luck? It was a pure fluke, right? So immediately, one thing you can say straight off the bat is that they could just do, increase the number of plants in each group and do replicates. The reason is, well, reasons are really, is one, you can identify anomalies. It will increase precision, right? As well as that, it increases the accuracy of the mean. Because if you're trying to work out the mean of like 3 things, if I have like, 10, 100, and then 6, right? If I work out the mean for those 3, so 100 plus 10 plus 6 divided by 3, my mean is 37 roughly. Whereas if I have 10, 6, 7, 8, 9, 6, 7, and then 100 there, so 10 plus 6 plus 7 plus 8 plus 9 plus 6 plus 7 plus 100. How many numbers is that? 1, 2, 3, 4, 5, 6, 7, 8. Sorry, 1, 2, 3, 4, 5, 6, 7, 8. Yeah, 8. Then divide that by 8. Can you see how the mean is looking a lot more small than this versus this? That's a more accurate mean. Doesn't mean the real accuracy is how close you are to true value. Doesn't mean that we're really close to what the real value is. We just know that the real value for the mean is that. Just a real thing here, real quick. A bullseye, shoot an arrow. Bullseye, accurate, right? Here, it's not accurate. Whereas if I do a bullseye and I keep hitting the bullseye every time and my mate does it and they hit the bullseye, they're precise. Whereas if I miss the bullseye, but I keep missing it in the same spot, and also my mate does, then it is a precise thing here, but not accurate. So you want to maximize both of them. A precise experiment isn't a bad thing necessarily. It means you can adjust it if need be to get to it, be more accurate. So yeah, so then we got more accuracy of the mean. Also, what you should do is you calculate the mean. And the reason is because then you get a more representative sample. and it would be more representative for the treatment. Sorry, I should say. You can't just really say more representative. You need to say what it is for the treatment. It'll give us a better idea of, well, this is actually probably what would happen. Additionally, what you should be able to do in this sense as well to improve this and get more confidence is to do a t-test between the two different between the means to identify if the difference in means, the mean increase in height really, is significant. As well as that, you could plot bar charts and you could add SDs as well. So this is good because it measures variability, and also SDs are less affected by anomalies. So what I mean by this is that the t-test is when you're looking for the difference, you're testing whether a difference in means is significant between two groups. So for example, one group had a mean height of 100 and another one had 150. The difference of 50, is that significant or is it not? Is one significantly higher than the other? Correlation coefficient is to look at to see if the correlation is significant. And then the chi-squared is to look at whether the difference in expected versus observed, the difference is significant. Here, we're comparing the two means of two different groups, so we're going to use the t-test. SDs, the whole point of that is to look at how spread you are around the mean. So here's the mean for this, and a really, really good set of results would be the ones that don't spread too far. The highest is not this high compared to that. Whereas here, we've got loads of spread between the two of them. And the whole bit about overlaps is also important too, because if you've got, like, for example, let's say this is the treated, and this is A, and that's B. There's the mean, there's the mean, there's the SD for this one here, and there's the SD for this one. They're overlapping, so these results aren't significantly different in this sense, right? Unless you do a t-test as well, but you can see here, this has got less variability. This is highest, lowest, highest, lowest, right? There's a bigger range all around the mean. Sorry, this is supposed to be like in the middle of them, right? Whereas if I do a t-test and I've got these really significant difference results, right? Then that means they're going to be something like this. Can you see there's no overlaps? None of the results here are here. These are all actively like specific, actually lower than here. There's a key difference between the two. So that's quite helpful in getting good results. So if they do these things in this experiment, then they can get a more confident prediction. Because if you don't do these things, then how do you know you didn't get lucky? If I measure, if I have 10 people in a room and I give them chocolate and all of them shit themselves after eating the chocolate, I can't confidently say 100% of people will shit themselves. One, I have no control to compare it to, so I don't know if, they all just did shit themselves and it wasn't due to the chocolate. Two, I've only done 10 people. How the hell am I going to go and say that to everyone else? Three, I have no idea about how they selected them, this and that, blah, blah, blah as well, but they've already told you about the validity, which they've done. We're looking at things they can do with their results. So it's not about yapping about, oh, well, it could have been they could have used the same species, a leaf, other factors, blah, blah, blah. You need to actually list it and say it yourself. So now it says, cloning plants is also known as vegetative propagation. Identify 3 advantages of vegetative propagation in agriculture. So, I mean, one thing that's useful is that you have predictable yields. Like if you're growing your prized yam or whatever, then if you keep growing it the same, you'll always be fine. It's also easier to harvest. You'll have more reliable income in that sense as well. You can do it at any time of year as well. Another thing you can say is that younger seedlings are less likely to survive as well from sexual reproduction, whereas if you just do it like this, you don't have to worry about that kind of stuff happening. As well as that, you don't have to wait for them to germinate and this and that as well. And some crops, as well, remember, can be seedless, which is a bit of a tricky part as well. Lipids are an important group of biological molecules. Lipoproteins are roughly spherical-shaped structures that transport lipids in the blood. Here's a simplified figure of a section of the widest part of a library of proteins. We've got protein, we've got other lipids, we've got phospholipid molecule, chilling. So calculate the number of phospholipid molecules in the outer surface of the lipoprotein. Assume that the whole pattern of proteins in this is continued across the surface area of the whole surface of the lipoprotein, and use the formula surface area of a sphere is 4 pi r squared. Okay, so I can't really do this one because I tried with this jank ruler, it doesn't work. So what I'm going to do here is you find the total surface area of the whole molecule using this, right? Then what you do is you find the surface area of these proteins and take them out from the total surface area, and then what's left is your phospholipids, right? Then what you do is you divide each one by the surface area of every phospholipid. So you work out the diameter of each component, divide it by 2, and then 4 pi r squared it. If you do that, the answer should be apparently 141. But yeah, can't really do that one, Saz. Lipoproteins with fewer phospholipids and more protein in the outer layer are known as high-density lipoproteins. Lipoproteins with a large number of phospholipids but less protein are known as low-density lipoproteins. Use this to explain why lipids can increase the buoyancy of aquatic animals. It's because, well, here it's saying that lipids have, you have lipoproteins that have a large number of phospholipids but less protein are known as low-density lipoproteins. So if the question's saying that there's lipids that can increase the buoyancy, it must mean that they are lowering the density of the thing. So that means lipid is less dense than protein. That's what gives them the floating. If you're buoyant, you're able to float in water, which means you're not going to sink straight to the bottom. And that's all to do with either being less or equally as dense. Lipids have many roles in living organisms. Some are used as energy storage in adipose cells. Unsaturated fatty acids contain at least one double bond between two carbon atoms and so contain fewer hydrogen atoms. All lipids are insoluble in water, and that's a good thing, so I don't really want them to dissolve. So they need to be transported in the blood by lipoproteins. Cholesterol molecules increase the stability. They make the fluid mosaic membrane less fluid mosaic. It makes it more rigid. And cholesterol is also used to synthesize the steroid hormones and bile. It's just factually recalling them one stars, mate. Triglycerides are a type of lipid molecule that can be broken down using hydrolysis reactions. Use the structure of triglyceride molecules as an example and explain what is meant by hydrolysis. So hydrolysis is where you use water to break, and this is triglycerides, 3 of them, so 3 ester bonds. Lysis is breaking here. So then you get 3 fatty acids split from glycerol. Anyways, I hope that helped. Please like, share, and subscribe. Thank you, and see you next time.